Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is William Dewig from Business Review, and I'll be your host. It's our pleasure to have Phage Consultants with us today, who will be discussing phage contamination as a major cause of process failures. Today's guest speaker is Dr. Marcin Los, CEO of Phage Consultants. I'd like to welcome you to our webinar platform On24. You'll notice that this webinar is browser-based, so if you do disconnect for any reason, please just click on the link that you received via email to rejoin the session. In order to ask questions, you can send these in via the questions widget. Just type them into the box at the top left-hand corner of your screen and click submit. We'll allocate around 10 to 15 minutes at the end of the session to address any questions or thoughts that you may have. Please use the yellow help widget if you require any assistance, and you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. But now, please allow me to welcome Dr. Marcin Los.
Welcome everybody during my webinar. Thank you very much for joining it. Today I will talk about phage contamination as a major cause of process failures. I will tell you about what bacteriophages are, why they are dangerous in fermentation, where do they come from, what are the symptoms of phage infection, what are the consequences of neglecting phage treat. I will tell something about our company, and then we'll go to concluding remarks and question and answer session. So first of all, what bacteriophages are. Bacteriophages are basically viruses which infect bacteria. They show a few possible lifestyles, and among those there is virulent lifestyle, temperate lifestyle, and chronic infection. During virulent lifestyle, bacteriophages infect bacterial host, inject its genetic material into the host, and force bacterial cells to produce phage components, which then are assembled into mature bacteriophage particles. Mature bacteriophage particles are released from bacterial cell in the process of lysis, and daughter phage particles can infect next bacteria. In case of temperate bacteriophages, the lifestyle is much more complicated. They can develop in exactly the same way like virulent bacteriophages, but a fraction of phages can integrate its genetic material with the genetic material of the bacterial cell and be propagated to every daughter cell which is produced from the infected cell. Occasionally, some of those cells may get induced, and the prophage will continue its development, producing phage components which will be assembled into mature bacteriophage particles, which will be released in the process of lysis and can infect more bacterial cells in the environment. This, in case of next chronic infections, this lifestyle looks in a bit different way. Bacteriophages infect bacteria, and they immediately start to produce components of progeny viral particles. But in most cases, they do not lyse the cell during the process. They release bacteriophages in a continuous mode without damaging the cell. So it means that such infected cell is extremely productive. It can produce much more phage particles during its life than in the case of two previously mentioned lifestyles. Of course, released bacteriophages are able to infect next bacterial cells and increase the ratio of production of bacteriophages. So there are a few facts about bacteriophages, and there are some numbers associated with them. It is estimated that the number of bacteriophages on Earth is 10 to the power of 31 up to 10 to the power of 32. It is a number which I can write down, but I cannot really imagine it. So I just decided to do some maths to check how big the number is. And after assumption that each bacteriophage is 100 nanometers long, which is a fair assumption, I just calculated how long line you can produce out of those bacteriophages. And actually the line is 10 to the power of 13 astronomical units, which is the length of and distance of the Earth to the Sun. And actually this still don't tell me that much, so it is, I calculated that it's 10 to the power of 8 light years, which is 1,000 times longer than our galaxy. So it gives an impression how big number it is. There are some more numbers, like every second it is estimated that a number of phage particles equal to Avogadro number is initiating a new infection cycle. It is a few tons of bacteriophages every second. Hopefully none of them in your bioreactor. The size of bacteriophage vary from 20 nanometers to 700 nanometers. The genetic content is also very variable. From in small phages, it can be like 3,000 bases. In large phages, up to 700 kilobases, 700 kilobases, which is even sometimes bigger than the smallest bacteria. Each of us is a carrier of about 1,200 bacteriophage species. And in general, on Earth, phages outnumber bacteria in the environment about 10-fold. And bacteriophages are present everywhere where life exists and hardly exists, even in extreme environments. So the question is why bacteria are still alive, outnumbered by bacteriophages, and which are present almost everywhere. So yeah, they manage somehow, to coexist with bacteriophages, with their sworn enemy, for more than 3 billion years. And what we observe in laboratory is that bacteriophages are really the worst enemies of bacteria, and they are really devastating. So it is not so straightforward why they can coexist. But the main reason is that in the nature there is no monocultures. There are always mixed cultures of different bacteria. There are different consortia, which prevent bacteriophages from killing all the specimens of given bacterial species. So they are protected by some spatial separation or by living in a more complex environment. There are also, limited resources on Earth, and the number of bacteria is quite stable. Basically, the same number of bacteria is dying at the time when the same number of bacteria is dividing. So it means that bacteria in the environment, they are not very productive usually, and they cannot produce so many progeny bacteriophage particles as bacteria in the fermentation. So fermentation, from this perspective, is a very unnatural event. It is a monoculture with unlimited access to resources and without any competition, which is normal in case of natural environment. Now we are coming to the next point of the presentation, why bacteriophages are dangerous in fermentation. So in general, fermentation suffers from several different contamination issues. They are viral, including bacteriophages, bacterial and fungal contamination. And the most devastating of all are viral contaminations. It is due to the fact that bacteriophages and viruses in general, they have much shorter life cycles than their host cells. They produce much more progeny, and due to small sizes, they are very hard to stop. Some of them are very durable, and it is very difficult to clean up facility later on. In general, when you take a look at the literature and scientific literature, it seems that bacteriophage contaminations are it's a problem only in case of dairy industry, and that mammalian virus contamination in biopharmaceutical companies are much more frequently found than bacteriophage contamination in other industrial biotech or biopharma industry. But it is a false impression. The reason for that is that bacteriophage contamination are almost never reported to the public, so there is no scientific papers, almost no scientific papers about it, and also not that much information in the public domains. On the basis of our experience, I can tell you that the most frequent troublemakers in bacterial fermentations are T1-like or RTP-like bacteriophages. The second biggest troublemaker, they are T4-like bacteriophages and phages belonging to other Myoviridae. And other types of bacteriophages are really seldom found as a fermentation contaminants. In case of T1-like and RTP-like bacteriophages, there are some, let's say, general properties you can expect from this type of bacteriophages. Usually, they are very desiccation resistant. Sometimes the resistance is a bit lower. Usually, they are medium. They show medium to high resistance to disinfection agents. And when you take a look at the samples from environment, they are not the most common in environment. So there is quite strong selection for them during the bioprocess. They show usually very high virulence with short development times and relatively high burst sizes. And what is also quite disturbing is that they very easily adapt to facility environment, to fermentation environment, and they can make their life cycle more effective, and usually they increase the amount of produced bacteriophage and decrease the time needed for that over time if you allow for reinfection. Second most common group, they are T4-like and other Myoviridae bacteriophages, and usually they show medium to low desiccation resistance. They are not as durable as T1-like bacteriophages, so it's easier to disinfect them or kill them. They are the most common bacteriophages in environments. In environmental samples, there will be like 60–70% of all phages. There will be Myoviridae. They show very different spectra of virulence. Some of them are very virulent, some of them develop relatively slowly and give relatively low burst size. Sometimes they show very complex development programs with very flexible reactions to physiological state of the host, and this is due to the fact that they are relatively big viruses with relatively complex development program encoded in their DNA. Due to this fact, they show medium to low probability of adaptation to the facility and the process. Other bacteriophages which are relatively seldom found in bioprocess, they usually belong to Leviviridae, sometimes Innoviridae, but also to Podoviridae. The members of Podoviridae show very similar behavior pattern to T1-like bacteriophages, but Leviviridae and Innoviridae usually they are they show very high to medium desiccation resistance, but they are extremely durable when it comes to disinfection and inactivation agents. For example, they are very resistant to UV light, which is caused mostly by the fact that their DNA is single-stranded, so it is less reactive than the double-stranded DNA. They are very rare in the environment. It's quite difficult to isolate them, so they are also not very common in the fermentation. And even though that they are extremely effective in utilization of the host resources. In general, due to very compact genomes and the fact that their proteins usually hold multiple functions, the probability of phage adaptation or evolution is relatively low in this case. Yeah, so next, this slide will show you why bacteriophages are so dangerous in the fermentation. If you cultivate a bacteria which is, let's say, E. coli, you expect it to divide every half an hour, which means that after 4 hours you will have like roughly 100 daughter cells present, which originate from one single cell. In case of bacteriophage, this number can be achieved just by a single event of infection within half an hour. If one of small bacteriophages is involved, sometimes the numbers are thousands of progeny viruses in less than 1 hour. So it is very easy and for bacteriophage to outnumber bacteria in the fermentation. On this slide, you will see the effect of the final stage of culture lysis, which was infected with bacteriophage. As you can see, there is no survivors, and all cells were bursted already. In general, in culture, usually some of them will survive, but usually there are some strongly mutated cells. So now we are coming to next part of the presentation. I will tell something about the origin of bacteriophages in the bioprocess. So where do they come from? In general, bacteriophages are very common in the environment, as you already noticed when I was talking about numbers of bacteriophages. And it is very easy to find them in the environment. But in some of bacteriophages will be easier to find in a specific type of conditions. For example, if you are running E. coli fermentation, it is not advised to place your production close to sewage, to highly contaminated rivers, or wastewater treatment plants, because E. coli is quite common in this kind of sources, and it's much easier to find E. coli phages there. In general, if bacteriophages are located, or rich source of bacteriophages is located in the proximity of your facility, it is much easier to contaminate, for example, processed water, especially if you use a river as a source of processed water, and also other raw materials, and not always it is possible to sterilize it properly. In general, moisture seems to facilitate the spreading of bacteriophage. So again, if you place your facility close to the river, you can expect that bacteriophages which are there, they will live longer. There will be some maybe aerosol created. Much easier then to bring them to the facility. So basically, the location of your facility is extremely important. But there is also, like a hidden enemy. If you run your process, you usually expect that it's outside environment, which is dangerous for your bacterial cells. But some of production strains can carry unknown or known prophages in their genome, and the effect of that can be as shown on this graph. The graph shows the effect of induction of bacteriophage lambda using different induction agents, and basically, when you look at the effects, you cannot really distinguish them from infection with the virulent bacteriophage. And the decision whether bacteriophage will get induced and upon infection will lysogenize the cell or lyse the cell is made by this simple molecular network, which is responsible for lysis or lysogeny decision of the bacteriophage lambda, and for this decision whether it will or will not be induced. In case of bacteriophage lambda, we can predict pretty well what will happen, but the majority of bacteriophages or prophages have unknown inducing agents. So if you have a strain with unknown bacteriophage or prophage in it, you may expect that the triggering factor for synchronous induction is anything. And even if prophage will not be triggered, there is always so-called spontaneous induction, which occurs during the replication of the cell, and which will cause that most probably in the supernatant of your culture there will be some bacteriophage particles present, which originate from the prophage. And when they infect bacterial cells, which do not have this prophage in genome, they will behave exactly as virulent bacteriophages. So you will have almost complete lysis with some survivors which will get lysogenized, and they may be resistant to the infecting bacteriophage, but not necessarily 100% resistant. There are some exceptions in this mechanism. So one may claim that it's the problem is with fully functional unknown prophages, but when you use defective prophages, which are present in many strains which were engineered using prophages, that you are safe if they are defective. And one of the most common prophage which is considered defective and is used in a production strain is the DE3. DE3 is basically the engineered lambda immunity 21 bacteriophage, which carry T7 RNA polymerase gene. And due to the fact that int gene, which is necessary for induction, was destroyed by the insertion of the RNA polymerase gene, it's supposed to be non-inducible and do not give any functional bacteriophages even when induced. But this is just a theory. The truth looks like this, that in the supernatant of DE3 containing bacterial cultures, you can see still that there are quite a lot of, usually quite a lot, sometimes there is bigger amount of smaller amount of those bacteriophages, which are sometimes even fully functional, like the one which is visible here. Those phage particles were able not only to lyse the cell but also to lysogenize. So it means that they could replace somehow destroyed int gene. This is very important to check whether your strain is able to release a large number of this kind of particles, because it can really vary from one strain to another depending on your product and the process. Sometimes there is a certain risk associated with this process which utilizes the free prophage, and it is very important to know beforehand that if the risk is high or low, and to take actions which can help you to mitigate it. It is something what we can help you in if you are interested. In general, prophages or prophages attack or release, they are not really dependent on the season or outside environment. However, in the case of virulent bacteriophages, the moisture is a very important risk factor. The majority of virulent phage infections, according to our statistics, occurs when there is an increased amount of moisture in the environment. And in temperate climate zones, we observe a sharp increase of phage contamination during autumn, and the second one, which is a bit smaller, during spring. In other climate zones, usually it is a wet season, for example, monsoon in India. So the question is how the moisture can influence phage contamination frequencies. There were several hypotheses about it, and some of them included also such factors as bird migration. However, there is no bird migration during wet seasons, especially during monsoon in India. So most probably it is just about moisture, not about any other factors. So moisture can stabilize some bacteriophages. Some phages are not really desiccation resistant, and when there is a lot of moisture in the environment, they can live longer. Another factor is that the moisture increases the risk of contamination carryover from outside of the facility. So, for example, on dirty shoes or clothes, you can bring some material from outside and then spread it in the facility quite easily. Another thing is that moisture makes air filter less effective. The higher the moisture is in the air, the harder it is to really get rid of tiny particles, which bacteriophages definitely are. So the question is how those bacteriophages, once that they get to the facility, how they can enter the fermentation. And there is no easy answers for that, because the primary infection usually do not leave any traces of bacteriophage penetrating once, for example, air filter or being able to enter the culture because of some tiny personal error. In general, the majority of primary infection cases which are able to attribute to a certain point, usually they are raw materials which were contaminated, because you can retest the batch of the raw material and find that there was bacteriophage and compare it with the phage, which was a troublemaker. In general, it doesn't mean that raw materials are the most frequent cause of the contamination, but it is the easiest to find out. Now we are coming to next part of the presentation. So, what are the symptoms of phage infection? Let's imagine that it is a growth curve of your bacteria, and if you have bacteriophage, which is let's say relatively fast-growing bacteriophage, it can very, very quickly take over the whole culture and then lyse it. As you can see here in the third hour of the cultivation, bacteria number of bacteria is already smaller than the number of bacteriophages, and usually it is the point when you can see that something very, very wrong is going with the bacteria culture. But there are also bacteriophages which show totally different behavior. They multiply very slowly in the culture, and sometimes they become evident on the very end of the fermentation, but sometimes you may not be aware of their presence and consider that the batch was successful and process it. So what are the symptoms of phage infections? In case of fast-growing bacteriophages, usually you have very rapid lysis, and which is accompanied by extensive foaming in most cases. Sometimes in fed batch culture, especially in fed batch culture, you can see that there is like a transient drop in the OD, but then culture is regrowing, but not necessarily. Sometimes also the lysis is almost complete. It really depends on a certain bacteriophage, what is its capacity to kill the culture. Usually you also observe the formation of aggregates. In case of slowly growing bacteriophages, sometimes you don't see any symptoms of phage infection. Sometimes the lysis is very late. Foaming may not occur even, and basically the phage may be present but not visible in the whole inoculation train. What is even worse, this kind of bacteriophages are very difficult to detect, and sometimes they may be present in your cell banks. Those phages can turn into fast-growing bacteriophages once you give them a chance to evolve, and after several cycles of evolution, they may turn into very, very bad, very quickly growing phages, which cause very evident lysis. So here are some numbers. The slowest growing bacteriophage we ever isolated from fermentation, it was 0.2 orders of magnitude per hour. And the fastest one which we ever isolated was over 5 orders of magnitude per hour. So you can see that the difference is very, very big. And here we can see the real data from fermentation, which was intentionally infected with T4 bacteriophage. And as you can see, when the number of bacteriophages, which is represented by green triangle, is growing, there are almost no sign of any irregularities until bacteriophage reach about 10 to the power of 9 phage particles per mil. Then you could see extensive foaming and the inhibition of the bacterial growth. Okay, now we are coming to the pool question, and I would like you to vote whether you or anybody in your organization ever experienced phage contamination. Could you do it for me, please?
Yep. So as Marcin's just alluded to, it is time for today's poll questions. Again, the question there is: have you or anybody in your organisation ever experienced phage contamination? And the potential responses are yes, maybe, and no. So if you could just select the option that is most appropriate to you, and then click submit. And Marcin, what are you expecting to see from the results of this question?
Yeah, I believe that like a majority of people know what phage infection is, so phage contamination is. So basically, I expect that a majority of people will say yes, or at least maybe. Some of people may just be interested in the prevention of contamination. So those people most probably will click no.
Brilliant. Thanks very much for that. Okay, so let's see how indeed the audience voted. Wow, and we can see here that no was overwhelmingly the largest response. What do you think about that?
Yeah, so it is actually like a good sign that people are interested in the subject, even though they didn't expect it, phage contamination. So it is like exactly opposite. Among my customers, usually they seek for help when something already happened, and we have quite relatively few, let's say, customers who want to do some prevention job to prevent the disaster. So basically, it's better, to be prepared than sorry. So I assume that it is good, good result. So can we go to another poll question?
Yeah. Absolutely. So it is now time for the second poll question of today's webinar. And the question is: if you have experienced phage contamination, when did it happen? And was it in the spring, summer, autumn, winter, a dry season, a wet season, or not applicable? And again, if you could just select the response that is most appropriate for you and click submit. And Marcin, I assume that you're expecting a certain kind of answer from this question as well.
Yes, my first guess would be autumn. Maybe like a spring and wet season will be like a second preferred option.
Okay, brilliant. And let's see. Yeah, let's see how the audience have voted on that then. Okay, and it seems that the majority of the audience have selected not applicable. Okay, so
okay.
Yeah.
Yeah, so I was not right, but actually, from our experience, definitely, it turned out that autumn is the worst season. Maybe people, due to the fact that there are our audience from many different spots all over the world. Maybe they don't have like a autumn like experience in Europe or in the United States. Okay, so let's go to next part of the presentation. So now we'll talk about the consequences of neglecting phage treat. So the first question is, is that hard to contaminate facility? Because when you come into especially biopharmaceutical facilities, they are nice, shiny, and you don't expect to find anything there. Some of them don't have bacteria inside, or hardly have any. But the answer is that it is extremely easy to contaminate facility with bacteriophages. You can, even without any symptoms of phage contamination in your process, you may have up to 10 to the power of 11 bacteriophages per mil. And if you spread just 1 milliliter of such culture, which is relatively easy during sampling procedures or some manipulations, you can when you spread those bacteriophages equally in your facility, you can contaminate 100,000 square meters with the density of 1 phage per square millimeter, which is 1 million phages per square meter. Yeah? And just to give you an impression how big this area is 100,000 square meters. It is the area of 14 full-size soccer fields. And when you do a harvesting, you may cause even much higher contamination, because it is relatively easy to generate during harvesting and downstream processing much of very heavy contamination due aerosol formation or some spilling. So downstream processing is basically the part of the process which usually contaminates facility the most. And if you increase the contamination of the facility, it's much harder than to clean it. And in case of some T1-like bacteriophages, if you don't destroy them, if you don't clean them properly, they will not disappear in time. So if you fail to disinfect the facility, they will still stay with you forever. Just some of them will stay with you forever, just waiting for opportunity to cause another outbreak. And to make the situation worse, majority of disinfectants are not effective against bacteriophages. And if you do not clean your facility properly, you will select for those bacteriophages which can survive longer in the environment, in facility environment, but also those which are more resistant to disinfectants which are not optimal. But then a situation is even much more difficult. So what may happen if you fail to clean the facility and to kill all bacteriophages which were there? So first of all, primary infection can turn into outbreak. If you release bacteriophage, and it will be able to penetrate your defenses and infect next fermentation, you will basically start an outbreak which is very difficult to stop, because you are increasing every time the contamination level. So if you increase your contamination level, then it is more difficult to recover, and it is going in circles like that. So you increase the contamination, you decrease your chances to stop it, and sometimes at certain point it is very difficult to break this cycle. In case of process, if you are not able to kill bacteriophage, you can create loops of reinfection which allow bacteriophage to evolve and to adapt to your process much better. So usually phages are then developing faster, giving higher progeny, and they are more resistant to disinfection. So another consequences of for process, especially for those processes which do not have to be fully successful to yield some products, like for example solvent production, is very low product yield. And basically, if you are not able to stop bacteriophage, the single infection or single contamination may turn into production paralysis. And the highest score among our customers was 10 months of total paralysis of the productivity before they ask us for help. So what to do to prevent bacteriophages? First of all, you need to think about it when you start development of your facility. You can really minimize the risk of the primary infection and make facility much easier to clean if you plan it in the proper way. Also, the development of the process is very important, because you can develop the process in a way which will make it less probable to get contaminated. You can even on the level of choice of the equipment, you can make better or worse choices. Some pieces of equipment are much more effective in stopping bacteriophages than the other. So another layer of defense is your standard operating procedures. If you construct them in the proper way and execute them properly, the risk is much lower. First of all, you need to remember that the personnel is one of the most important factors in stopping and spreading contamination. You should have in place emergency standard operating procedures, which will be initiated when the contamination is suspected. So this is very important. Otherwise, you have to reinvent the wheel while the contamination is going on, and most probably is spreading in your facility. To achieve that, you should know your enemy, and if you wish, we can help you in all these activities. So, when already contamination happened, what to do to troubleshoot them? Bacteriophages definitely are the most dangerous, so you should really check whether they are present in your fermentation. Sometimes it could be a failure from different reasons, but in majority of cases the failure is caused by bacteriophages. So if you are able to check it by yourself whether there are bacteriophages or not, please do it. If you are not able to do it by yourself, please send us a sample. We can check it for you. In general, you need to initiate some basic research to define strong and weak points of the contaminant, and this is something what we can do for you also. And very important is also facility and process audit, which will make troubleshooting much faster and will identify all weak points and all possible improvements which may stop bacteriophage and help you to produce a good short and long term solution how to stop and prevent bacteriophages. And now we are coming to the next part of the agenda. I will tell something about the company. So, Phage Consultants is present on the market since 2007. It is based on more than 17 years of experience of myself, and experience is mostly on bacteriophage development in bioprocess detection, rapid detection methods, troubleshooting, bacteriophage biology. There are 9 people involved in our activities. 6 of them are scientists and 3 of them are consultants. We have a laboratory, small manufacturing unit, and we are able to use some sophisticated equipment like mass spectrometry or electron microscopy, which is equipment of University of Gdańsk or Pomeranian Technology Park. All of our scientists underwent extensive training in phage research, and 2 of them holds a PhD degree earned in phage research. So what we do? Basically, we do all type of research and manufacturing, which is somehow connected to bacteriophages. Due to our experience, we specialize in research-supported consulting, which is very effective, much better than consulting, because just like a plain consulting, because we always develop custom-tailored methods to troubleshoot and to prevent bacteriophages, all types of contaminations. It is not just a solution which we sell to everybody. It's just made for you. And so basically, there's a list of our activities. It is not a closed list. We do many more things than just like consulting, contamination detection, research, construction of rapid detection method. Just name it and ask us for help. We can do it for you. A few words about our customers. In general, we always keep the identity of our customers secret, even if they don't have NDA with us. Majority of they do. On this map, you can see that blue areas are the areas where our customers come from. So basically, they are located in North America, South America, Europe, Asia, and Australia. And the majority of them come from biopharmaceutical or industrial biotechnology sector. So something about our achievements. We so far troubleshooted more than 120 cases all over the world. And we also helped to smoothly introduce new products in industrial biotechnology by helping in prevention of bacteriophages. We helped 5 out of 12 biggest pharma biotech companies according to 2008 ranking. We helped to change production process in some biopharmaceutical companies which drugs were already on the market. And the volume of fermentation we troubleshoot will rank from a laboratory scale to 1 million liter. And some of our customers decided to give us references. So those 3 you can see here, we don't need to keep secret anymore. If you visit my LinkedIn page, you can see the whole testimony they gave us. But if you need some contact details, if you want to ask them for some information about us, I will be happy to provide you. So now we are coming to the very end of the webinar, so concluding remarks. And I would like you to take home a bunch of some highlights from the webinar. So first of all, the bacteriophages can be really devastating in bacterial fermentation. And the most common among them are T1 and RTP-like bacteriophages. They are the most commonly found in the bioprocess. Phage infection symptoms may include growth, variation, foaming, and aggregate formation. It is the most common symptoms which you can see, and if you see something like that, always expect it is bacteriophage. Increased moisture is always a risk factor. Often it is associated with the season. And if bacteriophages, if you are able to reinfect, the process will evolve and will get nastier and nastier in your production facility. So good cleaning is very, very important. In case of a need to speed up the troubleshooting, you can contact us, and it is something what we do on a daily basis, so we can lead you smoothly through the whole process. And I would like to now thank you for your attention and ask if there are any questions I can answer. So while you are going to ask me questions, here are my contact details which you can use then to contact us. Thank you very much.
Thanks very much for that, Marcin. Okay, audience members, it's now time for today's Q&A session. So if you do have any questions, first of all, thank you to everybody who has submitted questions so far. But if you do have any questions, please just submit those in the questions box at the top left-hand corner of your screen and click submit, and we'll try to get round to as many of those as possible. But for now, the first question that we've had in is, do you think that single-use technology will solve the problem with phage contamination?
Oh, thank you for this question. It's a very good question, and it is the question many people ask me. Basically, the single-use technology was intended mostly to shorten the downtime of the facility and to make the process easier to repeat. But basically, if you have something what is made of plastic, which cannot be autoclaved in place, which cannot be inactivated, it is quite dangerous from the bacteriophage perspective, from the bacteriophage contamination perspective. So among our customers, there are some who adopted single-use technology, and basically it didn't protect them.
Thanks very much for that, Marcin. Okay, so the next question that we've just had come in is, do you know about recent studies on the best disinfectants and procedures against phages in the industry?
Of course I do, because we do this kind of tests on daily basis for our customers. With the disinfectants, it is relatively difficult to say which are the best, because depending on the point on the globe, you have different disinfectants available, and even if they are produced by the same company and have the same brand name, sometimes there are differences in the composition. So what I would recommend is to test disinfectants, every time test disinfectants against even some standard bacteriophages, to check whether the disinfectant which is available in your country is equally effective as the one which is available somewhere else. But in general, there are several modes of actions of disinfectants which we are looking for when we are looking for good disinfectants. So basically, there are some cross-linking agents. There are some oxidative agents which are very strong. Sometimes you can rely on pH or different actions, but it is highly dependent on bacteriophage which you would like to kill.
Yep. Fantastic. Thank you very much for that, Marcin. Okay, so our next question is, are there specific process parameters that influence the presence of the prophage released from DE3?
Oh, thank you. Thank you very much for this question. It's a really good question. We studied that for several different processes, and basically, it seems that there are several variables in this subject. First of all, everything what triggers or make it easier to trigger SOS response will definitely influence the ability of DE3 to get induced, but to get released, usually bacteriophage require activation of some recombination systems and other systems of bacteria. So basically, it is very difficult to predict it, but it is something what can be checked in a relatively easy experiment, because if you mimic your process in a small scale, you can basically monitor whether bacteriophage is released in a large quantity or not. Thank you.
Thanks very much. Okay, so we've got a question here where somebody's asking about the most effective method to eliminate a phage. So would it be sterilization, hydrogen peroxide fogging, UV treatment, disinfectant, or potentially something else? Marcin, what's your take on this?
Oh, it depends really where is the phage located. For example, UV is usually relatively effective. However, it is you have to remember that UV light is effectiveness is falling down dramatically with the distance, and it will never work in the shadow. So basically, if you are looking for UV inactivation for example, of the room or like a production facilities, it can be quite difficult to achieve it. However, it is a very good supportive method. In case of sterilization, usually autoclavation is very effective against bacteriophages. However, there are also some exceptions depending on the host which you use. If you have a host which is thermophilic, autoclavation may not be sufficient enough. So basically, really, it depends on what kind of material you would like to sterilize or to remove bacteriophages from. You should choose a different method. For example, for large areas, the good disinfectant should be the most effective. In case of hydrogen peroxide for fogging, you have to remember that, for example, it was I think the research done by Steris that they assumed that every place which is narrower than 6 millimeters will not be effectively sterilized by hydrogen peroxide because the circulation of air is not good enough. From our research, it turned out that hydrogen peroxide alone may not be effective against some bacteriophages. So everything depends really. There is really a devil in the details. Depends on which mixture for fogging you use. You can achieve really dramatically different results. Thank you.
Thanks again very much. Okay, so another question that we've had is, what are the best forms of material transfer to use in an environment with known phage contamination?
Okay, so if you want to transfer your raw materials through a contaminated area, it also depends on the facility which you are using and the device which you are using. If you have, for example, pass boxes or you just bring everything through the door. But basically, I would recommend to use some several layers of, for example, some plastic, which can be easily removed or easily sterilized with cold sterilant. And when you are entering the clean area, you should sequentially remove it just to minimize the content of bacteriophage on it. Okay. Sometimes the solution could be better depending really on the situation.
Brilliant. Thanks very much for that. Okay, so now I believe we're moving into kind of the last two questions that we've had that we have time for. So thanks again, everyone, for submitting. The first of these is how can you detect a lysogenic strain? Is another way to detect lysogene without mitomycin? I hope I'm saying that correctly. And UV.
Yeah. Okay. So basically, mitomycin and UV will trigger SS response, which in turn can trigger a majority of lambdoid bacteriophages and some other phages. But in general, if you think about P2-like bacteriophages and other phages which are not really SS dependent, mitomycin will not give you an answer. UV actually is we never use UV for induction of the prophage. The reason for that is that there is a very, very narrow window of the energy which you should deliver to your bacteria, which will induce bacteriophage but not kill the host. So if you overdo it, you may get false negatives, and if you deliver not enough, you will also get false negative. So basically, we use a combination of several methods. Mitomycin C is just one of them, but also we look for bacteriophages released during switches in the growth rate. There is also bicyclomycin, which can induce some bacteriophages which are SOS independent. So you know, like there are several methods. It really depends on which host do you use.
Brilliant. And our final question of today's session is: how often do virulent phages switch to temperate phages, and back and forth as well?
Actually, virulent phages do not switch to temperate phages because they are totally different group of bacteriophages. Virulent phages are not able to form lysogens, and temperate phages are able to do that. But I guess that the question was how often they may get induced and perform a virulent cycle, and then go to temperate cycle again. Basically, it is you know, like, as many times as possible. In general, bacteriophage in the form of viron do not hold an epigenetic information about whether it's lysogenized on a host before. So basically, if you induce a prophage, it will produce bacteriophage particles which can infect the cell, and majority of cells infected will undergo a virulent cycle. Just a fraction of them will undergo lysogenization, and this is something what can last for like thousands of phage generations. So basically, there is no limits in changing between temperate and virulent cycle of temperate bacteriophages.
Fantastic. Thanks very much for that, Marcin. And that actually brings us to the conclusion of today's session. So I'd like to, first of all, thank obviously all the attendees for giving up their time to be here today, and also for submitting questions. And I'd also like to thank Dr. Marcin Los for what was a great presentation, and also Phage Consultants for sponsoring today's session. To the attendees, you'll receive an email shortly telling you how you can access the on-demand version of today's webinar, or you can access this through our website, which is www.business-review-webinars.com. Once this webinar ends, a survey will appear in its place, and we would appreciate it if you could take the time out to answer the questions provided, just to give us some feedback. We look forward to sharing further webinars with you, so please do keep an eye out on the website just mentioned, and follow us on Twitter at BRWebinars for daily updates, and join our LinkedIn group, Business Review Webinars. Thank you all once again, and I hope you all have a nice day.